Imagine for a second, you're looking at a plant out in a field, and it's just baking in this brutal midsummer drought.
Oh, yeah, the soil is basically turning to dust at that point.
Right, just totally relentless sun. And I mean, on a macroscopic level, we all know what happens, right? The plant wilts.
Yeah, it looks sad.
Exactly. But on a microscopic level, there is this frantic, literally life or death survival sequence kicking off.
It's a completely dire situation for the plant. I mean, to survive, it has to instantly slam its microscopic pore shut.
Just to trap the water.
Yeah, exactly. Just to trap whatever tiny bit of moisture it has left in its tissues.
And see, here's the thing that I find so fascinating to think about. How does a leaf actually know it's thirsty?
Right, because there's no brain.
Exactly. There's no central nervous system routing a pain signal like we have.
No, it relies entirely on microscopic biological switches.
Biological switches?
Yeah, specifically proteins called receptors. They just sit inside the cells, constantly sampling the environment, waiting for a chemical signal to basically flip them from off to on.
And today, we are going on a massive, exhaustive, deep dive into exactly how those cellular switches are wired.
Yes, we are.
We're looking at a 2026 paper from Nature Communications. It's by Maximilian R. Stammnitz and Ben Lehner, titled The Genetic Architecture of an Allosteric Hormone Receptor.
It's an incredible piece of work.
It really is. Our mission today for this deep dive is to understand the absolute nuts and bolts of a biological switch at the atomic level. Like, what actually happens when we take a microscopic screwdriver to, every single piece of this machine?
And the findings here, they really challenge a lot of our classical assumptions.
How so?
Well, receptors turn out to be far more tunable and modular and prone to evolutionary innovation than we ever realized.
Wow.
Yeah, and understanding this doesn't just explain how plants survive a drought. It essentially gives us the instruction manual for engineering our own custom biological sensors.
Okay, let's unpack this, because before we get to the mutant plants and the, 40,000 data points in this study?
Yeah, over 40,000.
Crazy. Before we get to that, we need to lay the groundwork for how these switches function in the first place.
Right.
And that revolves around a concept called allostery.
Yeah, allostery. It's sometimes referred to by biologists as the second secret of life.
The second secret.
Yeah, right after the DNA double helix.
That is a high bar.
It really is. But in the simplest terms, allostery is the phenomenon where a molecule binds to one physical spot on a protein, and that binding causes a shape change at a completely different distant spot on that exact same protein.
Okay, so if you're listening to this and you've ever set up like a smart home system, I like to think of it like a really complex dimmer switch.
Oh, that's a good way to look at it.
Yeah, like you press a button on the wall in your living room, and it doesn't just turn on a light above your head. The physical press sends a signal through the walls that subtly dims the lights in the kitchen and simultaneously closes the blinds in the bedroom.
Right. Action here creates a physical change over there.
Yeah.
That is a highly accurate way to visualize it. But just like a dimmer switch, this isn't a simple binary on or off thing.
Right. It's a spectrum.
Exactly. Biological switches have a highly specific dose-response profile. And in the paper, the researchers mapped this dimmer switch using what's known as a Hill function.
Hill function.
Yeah, it mathematically measures 4 crucial parameters of the switch.
Okay, let's break those 4 parameters down for everyone, because they basically act as the map for everything the researchers do next.
Sounds good. So first you have basal activity. In the math, we call this B0.
B0.
Right. This is essentially the background noise. It measures how much the biological switch is literally on, even when there is absolutely no signal present in the environment.
So going back to the smart home, it's like the faint, annoying glow of a smart bulb when the dimmer is pushed all the way down to 0.
Yes, exactly. So that's B0. Second is maximum activity, or B infinity.
Okay, so the opposite, the absolute ceiling.
Precisely. When the chemical signal is turned up to its maximum concentration, how bright does that biological bulb get?
Makes sense.
Then third, you have sensitivity, known as EC50.
EC50, what does that stand for?
It basically measures the exact concentration of the chemical signal that's required to hit 50% of that maximum activity.
Ah, okay. So essentially, how hard do you have to push the physical slider to get the lights exactly halfway up?
You got it. And finally, there is steepness. This is denoted by the letter N, or the Hill coefficient.
And what does steepness tell us?
It tells us the suddenness of the response. Like, does the light gently fade on as you move the slider, or does it suddenly snap from totally dim to blindingly bright?
All right, so just to track this, we have B0 for background noise, B infinity for the max ceiling, EC50 for sensitivity, and steepness for how sudden it snaps on.
Perfect.
Now, let's look at the specific biological switch the researchers focused on to actually map these parameters.
Right, the plant receptor.
Yeah, it's a plant hormone sensor called PYL1. And the chemical signal it's looking for, the hand pushing the dimmer switch, so to speak, is a stress hormone called abscisic acid, or ABA.
ABA, yeah. So when a plant is severely drought stressed, its internal ABA levels just spike.
Because it's panicking.
Basically, yeah. And those ABA molecules float through the cell and they enter this physical pocket inside the PYL1 receptor.
And this is where the physical allosteric shape change actually happens.
Exactly. Yeah. PYL1 uses a mechanism that biophysicists literally call a gate and latch.
I love this imagery. Picture PYL1 as a microscopic hand.
Okay.
And it has two physical protein loops acting as the gate and the latch. So when the ABA hormone floats into the palm of the hand, the gate loop swings shut and the latch loop locks in place.
That's spot on. And the consequence of that gate closing is the core of the whole biological response.
What happens?
Once it's locked, the outside surface of PYL1 completely changes its shape. It suddenly becomes highly, highly chemically attractive to a target protein called ABI1.
ABI1, what does that do?
It's a phosphatase. Basically, PYL1 reaches out, grabs ABI1, neutralizes it, and that specific physical connection is what fundamentally unlocks the plant's drought survival genes.
Wow. So Stammnitz and Lehner wanted to know what happens if they mutate every single amino acid in PYL1 to see how it affects that exact survival connection.
Yeah, every single one.
But to pull this off at scale, they used a method called GluePCA.
GluePCA, yes. The Glueable Protein Fragment Complementation Assay.
That is a mouthful.
It is, but it is an incredibly elegant workaround for a massive data problem.
How so?
Well, they needed to measure the physical binding strength between PYL1 and ABI1 across thousands and thousands of variations. So they took an essential survival enzyme from a completely different organism.
Which was?
Yeast.
Wait, yeast?
Yeah, the enzyme is called DHFR, and they literally chopped the DHFR enzyme in half.
Okay, so they attach one half of this yeast survival enzyme to our plant receptor, PYL1.
Right.
And they attach the other half to the target, ABI1.
Exactly. So now you have millions of mutant yeast cells growing in a dish. If the plant hormone ABA is added to the dish and the mutant PYL1 successfully grabs ABI1,
the two halves of the yeast survival enzyme are physically dragged together.
yes. They connect, they function, and the yeast survives and grows.
That is genius.
It really is. By measuring how fast the yeast grows at different hormone levels, they can perfectly calculate those 4 Hill curve parameters for thousands of mutant receptors simultaneously.
Because more yeast growth directly equals a stronger allosteric switch.
Exactly.
Okay, wait, I have to stop you there, because the leap in logic here feels huge.
How do you mean?
Well, we are using a chopped-up survival enzyme inside yeast cells to measure the biological reflex of a plant baking in a drought.
Yeah.
How do we know this highly synthetic engineered setup is accurately reflecting real, native plant biology? I mean, it sounds like we might just be measuring the Kirks of yeast.
That is a fundamental question in synthetic biology, and it's a very fair point. Whenever you use a proxy assay like this, you risk introducing artifacts.
Right.
But they rigorously validated the GluePCA method. They took a subset of these measurements and compared them to independent in vitro measurements.
In vitro meaning outside the cell entirely.
Exactly. Tests done in an isolated test tube with purified proteins. No yeast, no living cells.
Yeah, what did they find?
The statistical correlation between the yeast growth and the pure biophysics was an R value of 0.82.
Wow, 0.82. That's really high.
It is a remarkably strong correlation. It proves that yeast growth in this specific setup is an incredibly faithful proxy for the plant receptor's actual behavior.
Okay, that makes sense. The proxy holds up. So let's just do a quick recap here for everyone listening.
A good idea.
So we've learned that receptors use allostery to act as cellular switches.
Right.
And PYL1 uses this gate and latch mechanism to trap ABA and grab its target. And finally, GluePCA measures this whole thing by linking protein binding to yeast survival.
That's the perfect summary.
So here's a question for you listening. Think about that dimmer switch in your own home. What if changing just one tiny wire inside made its absolute brightest setting twice as bright? Keep that in mind as we look at the genetic map next.
Because that's exactly what they started finding.
Let's get into that master map. Because they tested over 3,500 different mutations to this single protein.
Yeah, the scale of the disruption they found is honestly staggering.
Close to 90% of the mutations altered the dose-response curve in some measurable way.
90%, which proves that the allosteric function of this protein is highly, highly sensitive to its exact sequence.
But what really stood out to me was the sheer volume of gain-of-function mutations.
Oh, yeah. This was wild.
Over 15% of all the residues in the protein, when mutated, actually increased the receptor sensitivity to the hormone.
Yeah, they identified 19 completely novel sites that increased sensitivity that no one even knew existed.
And as they mapped all these thousands of data points, they uncovered a biophysical law that I find absolutely fascinating.
The distance-dependent decay.
Yes. Can you explain this distance-dependent decay?
Sure. So if we think about allostery fundamentally, it is about mechanical information traveling through a physical structure, right?
Right.
The researchers mapped every mutation's effect on sensitivity, that EC50 value we talked about, against its literal physical distance from the hormone binding pocket.
And what do they find?
They found an exponential decay in influence. Specifically, there is a log base 10 or 50% reduction in the mutational effect for roughly every 13 angstroms of distance from the pocket.
It's exactly like dropping a pebble into a still pond.
Oh, that's a great analogy.
Yeah, the hormone is the pebble. The moment it hits the binding pocket, it creates ripples of allosteric information.
Right.
And those ripples fade in a mathematically predictable, exponentially decaying way as they travel outward through the physical jiggle of the protein's atoms.
Exactly. But as they analyzed that decay, the data revealed a massive hidden variable.
A hidden variable.
Yeah. When they looked at the 4 Hill parameters across all these mutants, they noticed they were frequently moving in lockstep.
What do you mean by lockstep?
Well, for instance, if a mutation caused the sensitivity to drop, the maximum activity almost always dropped right along with it.
Ah. So the dimmer switch wasn't just getting less sensitive to your touch. The bulb's maximum brightness was capping out at a completely lower level, too.
Right.
But why would 2 seemingly independent variables in the machinery break at the exact same time?
Because of that latent variable, protein stability.
Protein stability.
Yeah. A protein is a precisely folded 3-dimensional shape. It turns out many of these mutations weren't subtly tweaking the switch. They were simply destabilizing the core structure of the protein.
So it was just falling apart.
Pretty much. Nearly 75% of all the quantitative variance across the entire experiment was explained just by the protein core becoming less stable.
Man, it makes me think of a wobbly Jenga tower.
A Jenga tower. Okay, I see where you're going.
Yeah, like the protein core is the central column of the blocks. If you mutate those central blocks, the entire tower loses its structural integrity.
Right.
And when the tower is wobbling, every single other function degrades simultaneously. I mean, it can't hold as much weight, it can't reach its maximum height, it's just compromised across the board.
That is exactly what the data showed. But this leads to a fascinating problem for a bioengineer.
Which is?
What happens if you mathematically factor out that wobbliness?
Oh, interesting.
Yeah, the researchers applied a regression model to essentially erase the stability factor from the data. They wanted to isolate the mutations that genuinely changed the signaling logic of the switch without just breaking the underlying structure.
And when you clear away the noise of the collapsing Jenga towers, you see that the receptor is surprisingly modular.
Highly modular.
It's like a custom-built machine where specific physically distant parts control specific switch functions completely independently of one another.
Exactly. They found that the distal beta 4 to beta 5 and beta 6 to beta 7 loops, which are relatively floppy regions on the far outer edges of the protein, they almost exclusively tune the maximum activity, the B infinity.
Wow.
Meanwhile, a completely different region, the N-terminal alpha-1 helix, independently tunes the basal activity, the background noise.
But they also discovered some severe evolutionary trade-offs in this modularity, didn't they?
Oh, absolutely. There are 2 specific amino acids, E171 and Y50.
Okay.
The data showed that if you mutate them, you massively boost the sensitivity of the switch. It becomes highly responsive.
But wait, in nature, across millions of years, evolution hasn't made those mutations.
Right, it hasn't.
Why not?
Because mutating those specific spots completely destroys the overall stability of the protein.
Ah.
Yeah, you get a super sensitive switch, but the structural integrity is ruined.
It is a beautiful example of evolutionary compromise. Biology consistently sacrifices peak performance in one specific isolated metric to maintain the overall robust survival and stability of the entire organism.
Very well said.
I completely follow the evolutionary logic, but The physical mechanics of this modularity still trip me up a bit.
What part?
Well, you mentioned the beta 6 to beta 7 loop controls the maximum possible response, right?
Yes.
But looking at the protein structure, that loop is over 20 angstroms away from the actual binding pocket.
It is, yeah.
So how does a floppy outer loop sitting that far away from the action physically dictate the maximum strength of the protein grabbing its target?
It's a brilliant structural puzzle, and the answer comes down to a concept called entropic reservoirs.
Entropic reservoirs?
Yeah. See, we are trained to think of proteins as static, rigid Lego bricks, but they're actually dynamic, constantly vibrating, shifting molecules. And entropy, in the specific biochemical context, refers to the amount of chaotic jiggling or structural flexibility a particular part of the protein possesses.
Okay, so a floppy outer loop has high entropy because it's just jiggling around a lot in the cellular fluid.
Precisely. Now, when the PYL1 receptor actually binds to its target, ABI1, it costs thermodynamic energy to freeze those flexible jiggling loops into a rigid lock shape to secure the connection.
Oh, wow. I never thought about that.
Yeah. So if a mutation changes the baseline flexibility of that distant beta 6 to beta 7 loop before the target even arrives, it changes the total energy budget of the entire protein.
So that distant loop acts like a hidden battery.
Exactly, an entropic reservoir.
Yeah.
It stores conformational energy that physically dictates how strongly the main binding interface can hold on, even from 20 angstroms away.
That completely changes how I visualize proteins.
It's wild, isn't it?
Yeah, they aren't static. They are deeply interconnected energy webs. If you stiffen a spring on one side of the web, it inherently changes how much force the other side can exert.
Exactly. And this genetic map strongly suggests that if you are designing a custom synthetic biosensor and you want to tune its maximum capacity without destroying it, you shouldn't mess with the dense core.
You target those flexible outer loops.
You got it.
All right, time for another quick mini recap for everyone tracking this at home.
Go for it.
We just saw that allosteric effects fade exponentially with physical distance, like ripples in a pond.
Yep.
And we learned that protein stability acts as this massive hidden variable dictating overall receptor function. And finally, receptors are highly modular distant flexible loops can independently tune the maximum signal.
Perfect.
So, question for you listening. If you were designing a custom synthetic biosensor for a medical device, would you mutate the dense core to change its stability, or the flexible outer loops to tune its maximum signal?
Definitely the loops.
Definitely. And speaking of custom biosensors, that flows perfectly into the final and frankly most mind-bending segment of this deep dive.
Phenotypic innovations.
Yes, we aren't just talking about subtly tweaking a dimmer switch anymore.
No, not at all.
The data revealed mutations that completely rewire the entire logic of the house.
Yeah, in any deep mutational scan of this scale, you are constantly looking for the rare, extreme outliers. And they certainly found them.
What did they find?
They identified 15 specific mutants, which is just 0.43% of the library, that acted as complete inverters.
An inverter. Okay, let's trace the logic on this.
Sure.
A normal PYL1 receptor is an OF on-N switch, right? It does absolutely nothing until the ABA hormone arrives, and then it turns on to save the plant.
That's the wild type, yes.
But these 15 inverter mutants bound to the ABI1 target without any hormone present. They were stuck perfectly on-N.
Yes. But the moment you added the ABA stress hormone, they let go. They turned off.
They completely flipped the underlying biological logic from OF on N to on off.
They did, and it gets even stranger than that.
How so?
They found another 22 mutants, which is about 0.63% of the library, that created a band stop response.
A band stop. Okay, so band stop is an on-OF on-N switch.
Correct.
Meaning if there's zero hormone, the switch is on N.
Yep.
If you add a moderate, like Goldilocks amount of hormone, it turns OF.
Right.
But if you add a massive, overwhelming amount of hormone, it violently turns back on N.
It's incredible. And to understand how a single mutation can create such complex logic, we have to look exactly where these mutations occurred on the protein's physical structure.
Where were they?
Almost all of the inverter and bandstop mutations happen right at those gate and latch loops we discussed earlier.
Okay, let's zoom in on one very specific mutant they highlight in the paper. It's called H142W.
Oh, this is a great example.
Yeah, so in normal wild-type biology, the 142nd amino acid in this chain is histidine, which is a relatively small molecule.
Right.
But the mutation swapped it for tryptophan, denoted by the W, which is this massive, bulky, double-ringed amino acid. I think of this as the bulky bouncer mechanism.
Bulky bouncer. I love that. It's a very apt visual.
Right. Because when you substitute a small wild-type amino acid for a massive aromatic one like tryptophan, that bulky bouncer physically takes up way too much space inside the empty binding pocket.
And its sheer physical volume forces the gate loop to close prematurely.
Yes. So the plant receptor basically tricks itself.
Exactly.
It thinks it has caught a hormone because the pocket feels full, even when it isn't. The gate closes, the receptor changes shape, it grabs the target, and the switch is locked in the on-in position.
But then
the actual stress hormone, ABA, arrives and tries to enter the pocket.
And when the ABA molecule attempts to wedge itself into the pocket, it causes what biophysicists call a steric clash.
A steric clash.
Yeah, because the bulky bouncer is already occupying the space, the pocket is entirely too crowded.
Right.
The physical structural collision between the incoming hormone and the massive mutant amino acid creates violent physical stress.
Oh.
And the kinetic energy literally forces the protein complex apart, breaking the connection to the target and switching the entire system O-off-F.
That makes perfect mechanical sense. But honestly, it also sounds like an incredibly specific freak accident for a single plant protein.
It does sound like that, yeah.
Like a microscopic typo causes a bulky bouncer to crowd a room and reverse a switch. Is this actually a useful mechanism for understanding biology broadly, or is it just a weird quirk of PYL1?
It is far more than a quirk, and this is where the paper really expands its scope.
Really?
Yeah. The researchers cross-referenced their findings with a completely unrelated deep mutational study on a bacterial protein called the lac repressor.
Okay, bacteria.
Right. That bacterial protein has a completely different three-dimensional structure and a completely different evolutionary history separated by billions of years.
Billions.
But when mutated, the lac repressor produced inverter phenotypes at a rate of 0.35% and band stops at a rate of 0.20%.
Wait, our plant receptor rates were 0.43% for inverters and 0.63% for band stops.
Yes.
The frequencies are remarkably identical.
They are. The fact that the frequencies match so closely across vastly different domains of life, from bacteria to plants, it suggests that this is not an accident.
It proves this is a universal rule of evolutionary malleability.
Exactly. Allosteric proteins are inherently wired with the latent potential to completely invert their logic with just a single point mutation.
It's wild to think about. Just one final mini recap before we wrap up here.
Good idea.
We just learned that rare single amino acid mutations can completely invert the logic of a cellular switch.
Yep.
Inverters flip off-F on to on-off. Band stops create an on-off-off-on response. And this is all caused by steric clashes where bulky mutant molecules physically block the natural hormone.
Spot on.
So think about this, listener. What biological processes would actually benefit from a band stop switch? Like a biological alarm that turns off only at one very specific Goldilocks concentration, but screens on if the chemical is too high or too low.
It is a brilliant, ready-made mechanism for maintaining perfect cellular homeostasis.
It really is.
And if we synthesize the totality of this deep dive, that is the ultimate takeaway from this exhaustive map of 40,000 measurements.
What's the bottom line?
We need to stop thinking of receptors as rigid locks and keys. They are highly tunable, deeply modular, and shockingly evolvable machines.
Wow.
By mapping their genetic architecture at this scale, we aren't just understanding the biology of how crops survive droughts. We are writing the instruction manual for engineering custom biological sensors.
That's true.
We can use this precise knowledge to design precision medical therapies that activate only under exact biochemical conditions inside the human body.
It changes the whole paradigm of how we view our own cellular machinery. And it leaves me with one final thought, something to really mull over.
Good to hear.
Today we learned that a single microscopic typo, putting one bulky amino acid in the wrong spot, can completely invert the logic of a biological switch from OFNN to ONS. It makes you wonder how many of our own human diseases, or conversely, our greatest evolutionary leaps as a species, are simply the result of a microscopic steric clash, acting like a flip switch in the dark.